really want to now open up the stage for you guys to ask us questions, and we can try our best to answer as many of them as we can in the time that we have remaining. But so we've put, again, cards, I think, on all the tables. It's to make it more effective. It's actually better to write your questions down. We'll then collect those, and we'll start try to answer as many as we can. While my colleagues are picking those up, I'm going to maybe just throw out a first sort of general question to the panel to get them talking. But we talked a lot about excitement in the field right now. Woody talked about the recent board meeting at the Fox Foundation and how he was seeing some excitement. And I'd like to maybe ask the panel, when you look at the last 10 years of Parkinson's, where are you really seeing some of the most exciting areas of movement today? Maybe I'll start with Carly.
Okay, so I think some of the things we didn't touch too much today, because I think we heard a lot of exciting things already. Some other ways of looking at some of the information we heard that's also really important. My long-term goal is to identify someone who's at risk for getting Parkinson's disease before they develop the significant motor features of disease, and be able to give them a safe and effective treatment so they don't get Parkinson's. That's my long-term, that's what I'd like to have happen. And I think that, in fact, we're starting to get a little bit of traction using the same tools and the same ideas that we've been talking about for disease modification, moving that window back a little bit and starting to be able to say, we're really looking at populations to try to figure out who's at risk for disease. Can we identify people who might be at risk and come up with thoughts about how we might be able to intervene even before the symptoms of Parkinson's begin. So to me, that's so hopeful. It's something we've dreamed of, and I think we're actually getting a little traction there. Basically using the same things we've talked about, the genetic tools, some of the work that we've been doing here, looking at other risk factors for disease, and also recognizing that both the genetic forms of Parkinson's and the other forms of Parkinson's, which are probably a lot more common, seem to end up with the same kinds of pathways that are affected when you look inside of the brain or inside of the nervous system. And so the treatments that work for one are probably going to work for another. So again, that's very hopeful, because it means we're not looking at everything being slightly different for every different cause of disease. But if we can get some of these answers from this direction or from that direction or from all directions together, we're going to be making steps forward in terms of being able not only just to stop progression, but hopefully maybe to prevent disease. So to me, that's very hopeful.
But I would like to essentially echo what Carly's been saying. I have a slide when I describe in general what I do, and I have a house that's on stilts. And then I take away one of the stilts, and then the house starts moving. And I use that to say that we know that at some point something happens where some of those stilts get taken away, and the house becomes unstable. And in my mind, some of the ways that we've been able to put names on those stilts is to use some of the genetic identification of molecules that may be disrupted in Parkinson's disease. So we're starting to put names on those stilts so we can find ways to put them back. And before, when they didn't know there was an environmental component, or maybe an environmental component to what may cause Parkinson's, now we have genetic components and environmental components, and now we can not only do gene-gene associations, but also gene-environment associations. And actually, Carly's group had a very nice publication on this with that alpha-synuclein target, where there's a gene-environment interaction. And for me, it's putting names on these stilts, because if we want to develop a therapeutic, we have to have targets. And it's the identification of those targets that has, I think, accelerated the field. And even, I've been working on this molecule LRRK2 for about 5 years now, and there's over 700 publications on this target. This is a phenomenal amount of work. And it's because as soon as we put a name on that stilt that was gone, we can start putting it back in place. And so for me, I think that's one of the biggest leaps, is the ability to use the technologies of genetic genome-wide association studies. We got 23andMe in the back. It's a phenomenal resource to be able to start associating what mutations we may encounter and the onset of disease, so we can use those as places to begin, so we can have new ideas and new ways to target disease therapies.
And I think the issue, like anything else, is a numbers game, right? As I look across the therapeutic development of Parkinson's, I'm enamored with the fact that there are multiple things going on at the same time, right? In any area where the success rate is not high, you need, and I guess the hockey analogy is you win the game by having more shots on goal, because only one in, very few actually make it. So I think that's an optimistic thing in my front across different areas. And I also think if you look outside of Parkinson's, the impact of genetics can't be understated, right? The personalized therapies in oncology and cancer based on the genetic alterations in tumors, et cetera. The development of therapies, promising therapies in Alzheimer's, there's a time lag between that, right? So some of the genetic discoveries that were made in Parkinson's about 10 to 15 years ago, or now about 8 to 10, are now just now reaching fruition because the process takes time. And I'm quite optimistic that therapies directed because of those genetic observations and tested in patient populations that harbor either those mutations themselves, et cetera, are going to increase our likelihood of being very successful in potentially modifying disease. So some of the key components, from a scientific perspective, are coming into place in Parkinson's, which I know Dr. Tanner said, 20 years ago did not exist, and it was very much, groping around in the dark. I think there's more light being shined on this area than there ever has been before.
So we have tons of great questions, so hopefully we'll be able to get through as many of these as we can. I'm going to try to bundle some of them because some of them are kind of overlapping questions, and so I apologize if we don't ask your specific question in the exact way that you asked it, but hopefully for time we'll be able to get at least as many conceptual questions answered as possible. So why don't we start first? A number of people asked again about this sort of participation in clinical trials, ways to do that. I mentioned Fox Trial Finder obviously is a new tool that can really help you to identify trials that might be in your area. So I invite all of you to go to that website and take a look.
Last thing about clinical trials is that for people who are local here, at the back table, Grace Liang is sitting there, Dr. Liang and I, both are involved in more than 28 trials here at the Institute. So if you're curious about your personal participation and you live nearby, you can stop back there too. So, yeah.
And Brian, just want to let you know, this is an area that I can't understate how important it is in accelerating new treatments, is to try to reduce the time and the cost it takes to take things through clinical trials. This is clearly the most expensive part of the process for industry. And this is an area where all of you can play a very active role, all right? And there are a number of different trials. Not all of them may be testing a new medicine. If you're hesitant to do that, there are observational trials like the PPMI, there are genetic trials. There are many different ways that you can get involved directly in trying to accelerate that. And it's something that you can take an active participation in and would be highly valuable towards moving therapies forward.
So it's while I had you, so there are a number of other questions people are asking that kind of broadly fall into this category of sort of what's sort of the logistics behind a clinical trial? How do you sort of make decisions along the way? What happens when a trial fails? Sort of general questions like that. I wonder if maybe you could walk us kind of through the anatomy, if you will,
Sure.
From a company perspective in particular. Of
The clinical trials, let's say, and I've designed and helped conduct many of those, are really a partnership between the clinicians, the doctors themselves that run this trial, and the sponsors that help to design it. But the key interface are your physicians that you interact with. So I encourage you strongly to speak to your local physician. You have a neurologist perhaps that is caring for you, et cetera. And, do sign up for things like the Trial Finder, become as educated as you can, but ultimately when you engage down that process, it starts with a conversation with the clinician who's running that trial at that site that you may be involved with, all right? They are usually very highly talented and experienced sites. They will certainly explain everything to you prior to participating in anything. One of the key options is you can always opt out. You can always say, that may not be for me, but it never hurts to engage in a conversation and find out, what it might entail. The trials themselves, usually, at least for the interventional trials, those that are testing a new medicine, you really only want to participate in one of those at a given time because you do not want to confuse things by perhaps being involved in more than one. It's very, very important to follow through with the particular procedures that are, set forth in the trial, and they've been designed specifically so that certain very important questions can be answered and bring a therapy to the regulators that will then be approved. And lastly, and this is an area where we have to work much harder, I'm quite disappointed, is the results of the trial, okay? Unfortunately, historically, when a trial was successful, certainly everyone heard about it. But when it was not, that was often never very well communicated. And I think that one of the big pushes that I personally do, and I think that industry is moving towards, is sharing some of that data on trials that have failed, and making sure that you have the ability to go back to your physician and get a really informed understanding of why that trial failed, because you put a tremendous amount of effort to be that. You deserve that feedback at the end, and I think we're working more towards that as well. But I think the key is the clinician, like Dr. Tanner, like Dr. Liang, are the key people that you interact with, and they will keep you certainly informed, so you know at each step in the process.
There were a couple of related questions about sort of the duration of trials, like how long is it related? So if something is in the clinical trial stage, realistically, assuming everything comes out positive, what may be leading the phases of trials? Okay,
so the earliest phases, like the phase 1 types of trials, are usually quite short. You're usually only involved for a few weeks' time. Their goal is to try to, depending upon the therapy, but to get some very basic understanding, not about whether it's effective, but where it might distribute in the body, whether it's initially safe to be taken or not, etc. And if things successfully make it through those initial phases, and the actual therapy itself looks like a viable approach, they often enter into a hypothesis testing or an early efficacy testing. This is usually the phase 2 trial. The number of individuals that are involved increase. They go from the order of a few dozen to usually in the order of close to 100 or 150 or so. Okay? Those trials usually last a few months. Okay? And once again, if something is successful through that second phase, they usually enter these larger and longer phase 3 trials. Okay? In my last job, I was overseeing a number of those, and they involve hundreds of patients, 400, 500, 600 patients in a trial. Okay? And they can last for a year to a year and a half, because oftentimes it takes time is an important component to see whether a therapy may be effective. All right? So there are different trials depending upon your interest at what level and how much of a commitment you can make. But the process itself, coming back to my earlier comments, takes many years and is quite capital intensive, right, to go through that process. But you can see that there are multiple avenues in the different areas where you could certainly play an active role, right? From the earliest stages where there may not be much information about the intervention, and you'll be watched very, very closely, to the later stages, which are often, are not done in the hospital setting at all, or done at home, where there's more information about the molecule, particularly about how safe it may be, and that there really is a seeking efficacy there. So there really is quite a range where you can become involved. And once again, you're a neurologist, the clinical trial sites can help explain that in much further detail.
And again, you kind of, I think, reiterate this point. A lot of the timeline of a clinical trial is, is sort of set by the trial duration. They want to treat you for a certain period of time with the drug to see the effects. But a good portion of the timeline of trials, again, is the enrollment period, more the logistics of the trial. And this is where a very fast, rapid enrollment into a clinical trial could just accelerate moving that trial forward much more quickly than it would normally. So again, it's a real call out for participation. So one last just clarifying question on that before we move away from clinical trials. Can you be in more than one trial at a time?
Typically for an interventional trial, testing a new therapy, you only want to be in one of those. Okay? But that does not necessarily, and each trial is different, but it doesn't necessarily preclude you from being in an observational trial. So you might be able to, at that same time, get genetic testing done, even though you might be you might be able to get, usually the trials involve staying on your current standard of care, therapy. They usually don't require you to remove any of the existing treatments you're on. Okay? But typically you would participate in a single interventional trial at a time. And once again, always, as they end, you can, if you're really quite interested, after a certain time, re-enlist for another trial. It's sort of thought military-like to re-enlist into a new trial. But yes, you certainly can become involved in various different types, not just interventional trials.
So some questions about potential other kinds of treatments that we didn't really discuss today, but things like exercise, sort of nutrition, some of the, things like coenzyme Q10, some of these natural supplements. What's sort of the evidence that those can be helpful and beneficial for Parkinson's? Carly, you want to start that?
Well, exercise is a win-win. I think there's increasing evidence that suggests that people with Parkinson's disease who are involved in exercise are sort of maximizing the options. It doesn't make the disease go away, but it certainly gives you the best shot at being your very best given your stage of disease. And there's really no stage of Parkinson's that you can't do some kind of exercise, even if it's, you're not able to get up and move around safely. You can still be involved in certain kinds of strengthening exercises. We found in terms of speech, and this is actually something Fox is involved in, that using your voice, singing, doing things like that is very beneficial. So there are exercises for the voice as well as exercises, for motor. Being involved in some of the kinds of classes, for example, like we have here, but there are also other centers that involve combinations of different disciplines. So yoga and Tai Chi, as well as the more aerobic exercise, will help with gait and balance, and will help to prevent against falls. So there are many, many things that you can benefit from in terms of exercise. In terms of some of the other types of interventions, so far we've been still challenged with coming up with things that have helped, that have been in the sort of non-pharmaceutical arena, like coenzyme Q10, which looked very hopeful in some of the earlier phase 2 trials. In a bigger phase 3 trial, turned out not to look as promising. So I think there's a lot of interest in pursuing these, but like all the other trials we've talked about, so far we haven't really hit a home run on that. If you want to comment more there.
Yeah, I think that's very much the case when it comes to supplements and things like that. My general view, I'm a neurologist, is it's really a safety issue. If when you speak with your physicians, if you're interested in nutritional supplements, have that discussion. If they are generally safe, I'm much more, aligned to actually engaging and going down that path, right? And like anything else, a lot of it has to do with how much you take and what interactions. So it's really a conversation with your physicians about that before you decide to go down some of those paths, right?
I'll just add that there is another trial that we're involved in that's still ongoing, which is a very, very long trial that's funded by NIH, looking at creatine, which kind of works on the same energy systems as coenzyme Q10. And that trial isn't completed yet because of some of the things we've talked about. It's going on for 8 years or more, which is much, much longer than a company could afford to support. So there may be an answer from that at some point, but we don't have the answer yet.
Could one of you talk a little bit about deep brain stimulation a little bit more? We sort of alluded to it. Maybe you talk a little bit about sort of who is kind of the right candidate for that, at what stage, and also maybe a little bit about what is DBS good for versus what it's not particularly.
So deep brain stimulation, as many of you may actually know or haven't, is a very tiny little electrode is put into a certain spot in the brain, and there are a couple of different spots that are selected for various reasons. And there's an external kind of programming device that allows the physician to pick settings that affect the brain transmission from one spot to the next spot in the circuitry that's involved in Parkinson's disease. So transmission in the brain essentially is electricity, and what the DBS does is it kind of helps to remodulate that, to reset the electricity. People who are good responders to treatment but have sort of rapid plunges going from high, lots of dyskinesias and then being really off and not able to function are classically the people who are the best responders to DBS. And often for people like that, the DBS can regulate your response so that you have effective motor function. You can take a lower dose of medication. You're not having these big peaks and valleys. And the dyskinesia can be minimized or in some people even absent. I have a patient I saw not too long ago who's one of my poster children for the best outcome of DBS, and he was a person like that who now is taking only once a day. He takes his anti-Parkinsonian medication, and before he had DBS, he was taking it 6 times, 8 times, and being up and down, and very dysfunctional. So that's the best outcome from DBS. There are side effects from DBS, and there are certain groups of people who do less well. Generally, people who don't have that kind of really good response to dopaminergic therapy are not the ones who do as well. People who have atypical Parkinsonism may not do as well. People who have cognitive impairment may not do as well and may actually even be worsened by DBS. So there are a lot of caveats, and that said, research is ongoing. The approaches to DBS are getting better. People are getting better at figuring out which constellation of symptoms responds to which spot in the brain. And so it is getting better and better, and I wouldn't want anyone not to be assessed by an expert in DBS to see if they might be a candidate, because it is increasingly, more people and more people can benefit from it.
Just a comment, this is a good example from, let's say, a development of new therapies approach. Here was a therapy that was developed in academic settings, and involved a surgical procedure, right? So this involves what's known as a stereotactic neurosurgical procedure to implant the electrode. And now because of the development of this therapy, more and more information about that procedure and the safety of that procedure was gathered. And that opened up the ability for the use of those stereotactic injections to deliver novel therapies across the blood-brain barrier, which is a challenge in CNS. So the Fox Foundation, there's a number of trials now that are using that basic principle that was applied in Parkinson's disease through DBS to deliver gene therapy. And there's a couple of trials that are ongoing to do that. And that avenue was only opened up because of all the work done with deep brain stimulation to show that you could safely do that surgical procedure in a Parkinson's population. So here's an example of sort of the collateral benefits of the development of a therapy and the opening up of new therapeutic approaches that are now being tested that are very, very similar to DBS but involve biological components instead of electrical.
So someone was asking, we talked a little bit about cognitive dysfunction, some of the non-motor symptoms of PD, but one thing we didn't really talk about is depression in PD, and how do you treat depression in PD today?
People with Parkinson's disease are, most people with Parkinson's are probably a little bit more prone to depression than an average person. Depression is pretty common, over the course of a life anyway, but people with Parkinson's chemically probably a little bit more prone to depression. Lots of times the depression in Parkinson's can be tricky to figure out because some of the features of depression can overlap with features of Parkinson's. So, the facial expression may not be as animated. People may not move as much. That can be PD, or it can be depression, or you can be a person who has Parkinson's disease and also is depressed. So one thing that's important is to be aware of it as a possible aspect of the Parkinson's set of symptoms, and know that feeling sad, feeling down in the dumps, and, not being able to motivate yourself can be from depression. Depression in Parkinson's disease can be treated with the same kinds of drugs that people who have sort of ordinary depression outside of Parkinson's also respond to. So the really important aspect about knowing about depression in Parkinson's is knowing that it can be treated. A number of the different antidepressants that are available now are effective in treating depression in Parkinson's. And that can make a big difference in terms of not just the mood of the person with Parkinson's, but also general function improves when you're not depressed. So other aspects of your symptoms, like your movement function, may also be improved if you take an antidepressant. So very important that the doctor assesses you for that and that an intervention with an effective therapy happens.
So a couple more great questions here. I thought, since we have Carly in the room, could you talk a little bit about sort of epidemiology in Parkinson's? So, people have heard if you smoke, you have a reduced risk of Parkinson's, and you alluded to some of your work about solvent exposure and how that might increase risk of Parkinson's. So you talk a little bit about what are sort of some of the current environmental factors that people have identified related to PD.
Sure. Okay. So some of the work that we do, which is very, very hard to do, because we're basically looking at people's life experiences and trying to understand which things in their life might be related to causing Parkinson's. And as I mentioned very early on, the reason we got interested in this was by the observation that found at this institute, which is that a group of people who were exposed to a particular chemical developed Parkinson's in a pretty short period of time, but it otherwise looked just like Parkinson's and affected the same brain areas as are affected in ordinary Parkinson's, where we're not sure of the cause. And that chemical was called MPTP, and it looked like another set of chemicals that are pretty common in the environment, for example, the herbicide paraquat. And we found we were able to go into the laboratory, take that chemical, go into the laboratory, and figure out that it affected the energy storehouse in the cells called the mitochondrion, and worked in one particular spot there. And that some of the things that also block that spot of energy production are also pesticides in the environment, like rotenone. And so we were able to take that observation in humans, go into the laboratory, figure out what this chemical was doing, then go back out into human populations and say what other things might people be exposed to that could be causing Parkinson's. And we've been working on this now for 25 years. It's slow going because you need a very different kind of information. That said, we've been able to identify a number of things that increase risk for Parkinson's disease. So, for example, the two chemicals I just mentioned, paraquat and rotenone, earlier this year we were able to do a study in farmers and actually identify that both of those chemicals in humans do increase the risk of Parkinson's disease. And surprisingly, this is really the first time that that association with rotenone has been shown, because rotenone is something that's pretty ubiquitous, can be used in all kinds of things, and it's hard to know who's been exposed or who hasn't. So we've been able to show that certain pesticides increase the risk. We recently published a paper that showed that exposure to certain solvents that are fairly common in industrial use, for cleaning equipment, but also are present in the groundwater. So the most common sort of contaminants of groundwater also are associated with an increased risk of disease. So again, we got this by getting very detailed occupational histories, working with occupational experts who could be sure that this job task caused that exposure, and showing that those solvents were associated with a greater risk of Parkinson's. And those also in the laboratory create damage that is in the same areas as are injured in Parkinson's disease in humans. So it's this back and forth all the time between the human population into the lab, from the lab back to the human population to get the plausibility for this. And then ultimately we hope for treatments. Some of the things that have also been shown over and over and over again are, for example, in the protective, what we think is the protective way, people who are smokers are much less likely to get Parkinson's disease. This has been shown since the 1950s, when it was sort of discovered accidentally, over and over and over again in human populations all around the world. Not that much work done in the laboratory on this, but a little bit that says, yeah, that does seem to make sense from looking in the laboratory. But another trial that is just now starting to take place that Fox Foundation is funding the U.S. component of, and the German government is also funding a component of this in Germany, is looking at nicotine in early Parkinson's to see if it slows progression. And our site will be involved in that trial, which is just about to get launched. So smoking, maybe caffeine, and again, this leads into some drug studies that people are thinking about doing that work in the same areas of the brain that caffeine affects. So maybe people who have greater exposure to caffeine are at lower risk. I've been involved in a study that, again, Fox funded, that was in China, looking at components of green tea. And we have tantalizing, not definitive evidence that there may be something going on there with some of the traditional medicines in China that may be affecting disease progression. So those things come from observations of human behavior. Exercise is another one. There's a little bit of evidence that suggests that exercise may be protective against developing Parkinson's disease. Another thing that is a risk factor is head injury. So people who have had traumatic brain injury also seem to be at greater risk for Parkinson's disease. All these things I mentioned to you as risk factors, and there are a few others, but solvents, pesticide, head injury, not everybody who has those exposures gets Parkinson's disease. And so one of the things we've just started identifying are the interaction between people's genes and their experience or their environment, and how if you have a certain genetic substrate, you're at much greater risk of having the adverse outcome, Parkinson's disease, if you have an environmental exposure. So we recently published a paper where people with a certain genetic variant in the alpha-synuclein gene. So this is a very rare cause of genetic Parkinsonism, but there are a lot of variations in the gene that don't actually cause Parkinson's, so you can say this person has it, that person doesn't, so it makes people a little bit different. People with a certain genetic variant in the alpha-synuclein gene who have head injury are 10 times greater risk of developing Parkinson's disease. People who don't have those genetic variants and have head injury don't have an increased risk. So that shows you that it's really, in most cases, going to be the combination of genes and environment. And so piecing that together is what we've been doing in our epidemiologic or population studies. We always collect blood, we always look at DNA, and we always try to do our analyses looking at combinations of those things. So bit by bit, we're starting to piece that together.
I think it's important, that type of data, when we opened this panel, all of us commented on the importance of a genetic discovery, and Dr. Nichols here, his life's work is working on a target that is now associated with genetics. I can't also appreciate how important the epidemiologic data are, because they point to potential targets for drug discovery, right? Because as you identify risk factors or modifying factors, they then shed some light on where you might invest some of your money in industry in developing a therapy. And some of that, might lead to new chemicals at a certain target, but sometimes they're just very, very straightforward. Like a recent observation that higher levels of uric acid in your blood may be protective, and there is a molecule, it's a dietary supplement that could raise uric acid. Industry is not interested in that because there's really, it's a for-profit entity. They can't necessarily make money off of a dietary supplement. The Fox Foundation has stepped in and is doing a trial to test the safety of raising uric acid, because there may be some downside to that. And before everyone would run and take this, we want to get a better understanding of, is it safe and might it be effective? So the epidemiologic work, although it's very, very difficult, is highly informative as well towards even the development of new therapies, not only clues and insights into the disease.
So I have to ask this, since this is California, any evidence for marijuana being protected for
Yeah, I will say
having systematic
that
benefit?
we just presented a paper at the American Academy of Neurology this year, actually, that was in collaboration with a colleague of mine who's an epidemiologist in Northern California, Kaiser Permanente, where we were able to look at information that people reported on a members survey in Kaiser, when they came in, so 20, 30 years ago. And we looked at people's self-reported use of illicit drugs like amphetamines and marijuana. The take-home so far, at least, is that it's not a good thing, and in fact appears to increase the risk. Both marijuana and exposure, either medically or otherwise, to amphetamine products and stimulants are each separately associated with an increased risk of Parkinson's disease. But we have a lot more work to do there, and I believe it will probably be just like everything else. Not everyone who used marijuana is going to get Parkinson's disease, and there's going to be a complex interaction between your own metabolism, maybe where you running marathons and using marijuana, you might be in better shape, right? So.
A couple of other questions. One, I wanted to actually have a question for Jeremy. PPMI and other studies are collecting a lot of clinical samples, biological samples. I wonder if you could talk a little bit about how, in your lab, you would use those samples to study Parkinson's disease.
Yeah, so this is, we're actually, because of the way the lab is set up and the way we focus our efforts, we essentially are geared towards trying to assess samples from patients so we can define whether the target that we work on can be actually used as a biomarker for disease. This target LRRK2 is one of those stilts, and one of the things that we're doing through funding with the Fox Foundation is to develop assays so we can see how much of this protein is around in cells, and also how it's modified, so does its activity change across the course of Parkinson's disease. And now that we have, and we'll tell Brian and everybody, we think we have this technology in place that it actually works. And so if we were to access these samples, we could say, assess the differences between controls and patients with Parkinson's disease, and then define whether this target could actually be used to predict disease onset, or maybe we could at least use this as a way to assess the progression of disease. And so not only does the foundation fund efforts to try to define those ways of detecting these molecules, particularly in patient samples, but they're also funding ways and standardized ways of ascertaining those patient samples. So I can envision once this grant gets done, we would potentially need to now try to apply our new technologies to the PPMI samples. And this is one of the integrated things that I think is beneficial about the Institute. The Fox Foundation actually funded us to find out the best way to procure those samples. And so these are actually being put in place with the PPMI. And so we have essentially an integrated effort with basic science and also clinicians that are ascertaining samples so that we can define biomarkers for disease. And so that's a long-winded way of saying we're going to take what we do in our lab and put them with the PPMI samples, and hopefully we can get a result that says we can track Parkinson's or not.
And from a development perspective, what you need to register a new therapy with the Food and Drug Administration is to demonstrate a clinical benefit, right? Usually on the core, some of the symptoms of the disease, and ideally improve quality of life or some ancillary improvement based on that therapy. What you do not have to show is a biological effect, okay? But more and more, and this is particularly the case in the LRRK2 instance, those biological effects are important indicators early on when we're investing in a therapy about whether it may work or not, okay? So in this instance, the ability of therapies in very early stages of development, they may not, in those smaller early trials I talked about, which are not large enough to show a clinical effect, but if we can show a biological effect, that we can modulate this, that, in quote, de-risks that program. It's much more likely to continue further on to the more expensive stages of clinical testing because you've shown a biological effect that's meaningful earlier on in disease. And I think even in the later stages of the trial, having as a primary outcome of a trial a clinical outcome, but having what we call a secondary outcome, being a biologic one, is going to be used more and more to support the development of new therapies because it provides additional evidence that you are having a desired effect. So I think that studies like PPMI and applying that to samples that are gathered, and often it's blood samples or CSF samples, is going to become a more and more important part of developing drugs for Parkinson's disease over the next few years.
Great. So we had a few more questions, but I think for time we may have to end now, because I know some people have to get up and leave. And I know some of the Fox Foundation staff, we have flights this afternoon. But thanks so much. Really, really great questions. Hope this was informative for all of you. It's a pleasure for us to be here and be able to present this information to you and to speak with all of you. I think some of us should be around for a few more minutes. If you do want to come up and ask the question that maybe didn't get addressed adequately, we're happy to try to address it now. But thanks so much, and enjoy the rest of your weekend.